C1, which is present in the serum, which is a macromolecular complex that consists of C1q, C1r, and C1s. It is combiningly known as C1q-R2S2, which is stabilized by calcium ions, is in which the C1 molecule, C1q molecule, is composed of 18 polypeptide chains, 18 in a pair of 3, that is 3 into 6. 6 you see as finger-like projections, and each is made up of 3 polypeptide chains. That makes the 18. The pentameric IgM is bound to the antigen on the target surface, and it assumes the so-called staple configuration. We will see this staple configuration in a moment. In this configuration, at least 3 binding sites for C1q are exposed. That is, when antigen and antibody, they bound, that when IgM is interacting in this antigen-antibody complex, the IgM assumes a staple configuration, that is in a staple-like. You have heard of a stapler, right? And the pins of that stapler. Now imagine the pins of the stapler. Similar configuration is visible with the IgM. Now the circulating IgM, which is normally in a planar configuration, in which the C1q binding site is not exposed, it will not activate the complement cascade. It is only the staple configuration that activates the complement cascade. You can see here the 2 configurations of IgM, that is the planar configuration and the staple configuration. This is the molecule of IgM in both its form. So it is the staple configuration that activates the complement because its binding sites are exposed for the binding of the IgM to the C1q molecule. Now, when in case if IgM is not there, if IgG is the molecule that is forming this antigen-antibody complex, it contains only one single C1q binding site. So firm binding is achieved only when 2 IgG molecules with a spatial of 30 to 40 nanometer of each other are present on the surface of the complex. That is, in short, we can say that because IgM is a pentamer, it's easy for IgM to activate the complement component, that is C1, but it needs more than one IgG molecule to activate the C1 component of the complement. That is, when 2 IgG molecules within the 30 to 40 nanometers of each other provide the binding site of the C1 component. This difference accounts for the observation that a single molecule of IgM, which is bound to a red blood cell, can activate the classical pathway and lyse the red blood cell, whereas almost 1000 molecules of IgG are required to assure that 2 IgG molecules are close enough to each other on the cell surface to initiate C1q binding. Now the first step is visual here, that is the binding of C1q to the antigen-antibody complex. You can see the antibody in the green and the C1q inverted form is visible. So once this binding takes place, that is C1 is bound to the antigen-antibody complex, the C1s component of the C1 complex, it cleaves C4 and C2. C4 is broken down into C4a and C4b. C2 is broken down into C2a and C2b. Now cleaving C4 exposes the binding site for C2. Now C4 binds to the surface and C2 binds to the C4, and this forms a complex, that is C4b2a. This is also known as C3 convertase. As the name suggests, this complex will act now on C3. So the C3 convertase that we saw in the previous slide, it hydrolyzes C3 molecule, and not just one but many C3 molecules. Now some of these C3 combine with that C3 subcomponents of C3, they combine with that C3 convertase, and this will make another complex. This will form another complex, that is C5 convertase. So that you can see in the visual below that C4b2 C2A binds with C3B component of C3 and forms the C5 convertase. Now you can realize the cascade occurring. The first C1 which activates C4, and then comes the C2 and now the C3 component. When C3 binds, it forms the C5 convertase. As the name suggests again, that this enzyme, this protein is going to act on C5 component of the complement pathway. Now C5 convertase, it binds with the C5 and thus cleaves it into C5A and C5B. C5A will diffuse away from this side, whereas C5B, which is the central molecule of this pathway, will have its function there itself. Now let's see what happens next. Now C5B, it binds with C6. Now there is no more cleavage taking place here. That is, C5B simply binds with C6. This initiates the formation of the membrane attack complex. That is, C5B will bind with C6, C7, C8, and this C8 will then bind with C9. So this poly C9 along with C5B, 6, 7, 8 is the membrane attack complex. Now once this membrane attack complex forms, you can say that the classical pathway of activation has stopped in a way. But the complement function hasn't stopped here. So this comes to the next point of discussion, that is alternate pathway. Till now we discussed the classical pathway. Now we are discussing the alternate pathway. Now in this alternate pathway, antibody are not required. No antibody is required. So how does it get activated if no antibodies are required? Now the alternative pathway basically is a component of the innate immune system. And this pathway involves 4 serum proteins. It involves C3, factor B, factor D, and properdin. This pathway is initiated by both gram-negative and gram-positive bacteria. So you can say that the gram-positive bacteria, gram-negative bacteria, when they come inside the body, when they bind with the cellular parts, or when antibodies bind with this antigen, when this antigen, that is gram-positive and gram-negative bacteria, this will initiate the alternate pathway. Now, as you can see in the next slide, the C3 which is present in the circulation, it gets hydrolyzed spontaneously. This is a spontaneous process, that is C3 will be converted into C3b and C3a. C3b fragment attaches to the foreign surface, that is, that antibody, that antigen and the antibody complex, which is a foreign surface, to that C3b will bind. Now factor B, it binds to the C3b component which is there, and it is acted upon by factor D. So you see that C3b is bound with factor B. Now this complex, that is C3b factor B complex, is acted upon by factor D. Now this factor D will cut a portion of factor B, okay, and this will form a formation of C3b and small component B of factor B, the large component of factor B, that is B small b. So a complex is formed, that is C3b capital B small b. This complex is actually a C3 convertase enzyme or a protein. It has C3b convertase activity. So it is this complex that now will act on C3 again. Now C3b capital B small b, this complex is stabilized by another factor, that is properdin. So the binding of properdin stabilizes this C3 convertase. Now C3 convertase will generate more C3b, that is, it will break down more C3 molecules into C3a and C3b, and some more C3b will bind to this already formed complex. So a second complex is formed, that is C3b capital B, small b, 3b. That are three components. One is C3b component. Second one, which is purple, is the small b, the b component of factor B, and the third component is again C3b. So it is like, you can say, a sandwich of two C3b's and the B component of factor B. This has C5 convertase activity, means it will act now on C5. So now C5 convertase, that is the complex which we saw in the previous slide, will cleave C5 to C5a and C5b, wherein the C5b will bind to the antigenic surface, initiating the membrane attack complex. So now both the pathways will converge into the membrane attack complex. The classical pathway also resulted in the membrane attack complex. The alternate pathway will also result in the membrane attack complex. Now, this further discussion will be carried out by Professor J.P. Patel here, and I hand over the work to him.
Thank you, sir. As two pathways of complement system, that is classical pathway and alternative pathway, discussed by Mansoori sir, and the third pathway also known as lectin pathway, that is we'll discuss over here. And in this pathway, the involvement of C4 and C2 is also there, especially complement pathway, classical pathway that involves C1, and alternative pathway that doesn't involve C1 and C4 and even C2, that start with C3. This pathway, another third pathway of complement system, that starts with a specific kind of molecule. Especially this pathway doesn't require presence of specific antibodies, but a specific kind of glucose or mannose molecules which are found on the surface of bacteria, and that is known as MASP, mannose like binding protein, or specific serine proteases MASP1 and 2. This play important role in the activation of the third pathway, and these are analogous to C1q molecule of the classical pathway. So this pathway, as we know that in lectin pathway, a specific molecule, this pathway originates when the host protein bind to the microbial surface, and these lectins are the protein that bind to specific carbohydrate, that is mannose. This mannose found on the surface of microorganism, especially on the cell wall of microorganism. This MBLS, that is mannose binding lectins, bind to mannose on bacterial surfaces of Salmonella, Listeria and Neisseria strains, along with this Streptococcus neoformans and Candida albicans. These are the other organisms where these molecules are found. This lectin pathway, also known as mannose binding lectin, that is MBL, binds to the foreign surface, and this is known as the protein known as MBL associated serine proteases 1 and 2, that is MASP1 and 2. This bind MBL and generates activated C1 like complex. So we can say that MASP1 and 2, that the function of MASP1 and 2 that is similar to the function of antibody, especially C1q molecule, C1q, R2 and S2, binding of MASP1 and 2 on the complement system that cleaves out C4 and C2. As we have already seen that C4 cleave into C4b and C4a, C2 cleave in C2a and C2b. The larger component of this C4 and C2, that is C4b and 2a, that bind with each other and form a complex known as C3 convertase. The substrate for this complex, that is C3, and they work on C3, leaves out C3 into C3A and C3B. C3A being a smaller component that diffuses away, and C3B a larger one that again bind with these complex, that is C4B, 2A and 3B, and that resemblance in the classical pathway. Then this C3 convertase that cleaves out C3 into C3A and C3B. This C3B again the complex that form number of C3 molecules, C3B molecules, the larger molecule that bind with the previous complex and they generate C5 convertase. And this C5 convertase that cleaves out C5 complement protein into C5A and C5B. C5A being a smaller component that diffuses away into surrounding, and C5B a larger one that binds with that attracts another components or the protein of the complement pathway. This C5B that attracts C6, C7, C8 and a form complex trimolecular complex C5B, 6, 7 and then 8 also bind over here. This the activity of each complement component or complement split product that is very short lasting. So if it is not stabilized, the active component of complement split product that become inactive, and that is very much necessary to control the activity of complement system, because along with the killing of foreign organism that also carry out some detrimental effect, harmful effect on the host cell of